and I'm really happy to present my talk on bryophytes and lichens of Texas, specifically focusing on some recent research that we've been doing at some properties up here in north central Texas, and also I'll speak a little bit about Mason County. This slide that I show here as a title slide, it turns out to be a really good slide for talking about lichens and bryophytes, especially in comparison to the vascular plant flora, the flowering plants, because there's a real kind of parallel between diversity of these groups, and albeit lichens are fungi and not related to vascular plants, we just know so much about the Texas flora and so little about the vascular plant flora, and so little about these bryophytes and the lichen-forming fungi. Again, Texas is rich in floral diversity. There's over 5,000 species. There's been documentation of the Texas flora going way back into the 150 years ago, and so albeit, and Jason and other members in this group can talk about new discoveries being made in Texas. We know a tremendous amount about the Texas vascular flora. We also know a lot about, some rare species and endemism. So this coneflower, the yellow coneflower in the upper right, the Echinacea paradoxa, it doesn't occur in Texas. I'm showing this picture because it does occur in southern Oklahoma. So are there potential relictual populations here? The cactus, the Acanthocereus, is something that's only found in the very southern tip of the state of Texas. And just some other examples of some endemic species. The images on the lower left are of a aquatic to semi-aquatic fern in the genus Marsilia. And there's two different forms of this one species, Marsilia vestida. This form on the left with the narrow leaves, which we found populations of at the Fawcett Wildlife Management Area in Palo Pinto County, potentially could be one of the rarest ferns in North America. Texas has some well-defined ecoregions. However, those boundaries overlap. And so, you go from southeastern coastal plain, kind of almost subtropical, but much more precipitation in the eastern part of the state, and you get drier and drier as you go over to the Trans-Pecos region in West Texas. And we know, because of our understanding of the vascular plant diversity, that plants follow these trends. And so, you don't expect to find plant communities to be similar to those from East Texas to West Texas, and a lot of times in between. There's not as much known about the bryophytes of Texas, but before I get into a little bit about the history of bryophytes in Texas, I just want to introduce you quickly to what bryophytes are. They're non-vascular plants, so these are plants that do not conduct water. They don't have roots, and they don't have a system of tubes and pipes, the xylem tissue that conducts water throughout the plant. They rely on the ambient moisture in the air and on the substrate surface. They include mosses, liverworts, and hornworts. The gametophyte is the dominant life stage, and so in a flowering plant, for example, the gametophyte in flowering plants are pollen grains, and you only see those during floral development, whereas in bryophytes, most of what you see, unless they're producing spores, are the gametophyte stage. Mosses come in two kind of morphological categories, acrocarpus and pleurocarpus, and I'll speak about that. I'll also talk about the two different main morphological forms of liverworts, thallus versus leafy, and then I'll briefly mention hornworts, the third class of bryophytes. And then after I introduce bryophytes, I'll talk a little bit about the lichen-forming fungi. So there's approximately 12 species of mosses worldwide. They're, in the tropics and subtropics, there's the highest diversity. However, for Texas, the most recent treatment for the state documents almost 300 species, but that's from 1954. McGill in 76 conducted a survey of the mosses at Big Bend National Park and found 109 species. And then we have some rare and or threatened species. Lorenztiella macroneuron, an aquatic species from the Edwards Plateau, is a species of moss that's received a lot of attention here in the state. if you go through the literature, you can find that there's approximately 350 to 375 mosses recorded for the state. So that's where we are with respect to mosses. On the lower left hand, I just showed a diagram of the difference between an acrocarpus and pleurocarpus moss. I'm going to mention a lot of terms today that you know, please look them up on your own. I definitely won't be able to give good definitions for all of them, but acrocarpus means that the sporophyte with the spores and the capsule at the end develops from the tip of the branches, and pleurocarpus mosses, the sporophytes develop from the lateral portion of the branches. And so, for example, the upper left Funaria flavicans, that's a acrocarpus moss. So that stalk is coming from the tip of a branch, and then the entodon below it is a type of pleurocarpus moss. These are two very common mosses that you can see throughout the state. Liverworts, there's approximately 8,500 liverworts worldwide. Texas has about 120 species documented for the state. The two groups of liverworts are thallose and leafy. The thallose liverworts are, there's 4 images on the left. So the Rabulia is probably one of the most common taxa in the state. If you haven't, you know, if you've seen something that looks like a liverwort, there's a good chance it's Rabulia hemispherica. It's very common, especially in the eastern half of the state. And so thallose liverworts have kind of a flat surface appearance, whereas the leafy liverworts on the right look like they have little overlapping leaves or scales. Rixia is, I just mentioned this, it's the most diverse genus of liverwort in the state. There's about 35 species of Rixia in the state. This is a small, sometimes called a microthallose taxon. And it's really common and diverse in Texas, and I'm going to talk just a little bit more about it in a moment. But one thing you note here too is that there are mosses. If you look, you know, if you look around there, you could probably find some lichens, and so these organisms do co-occur together. So when you see liverworts, not only are you going to see probably lots of other mosses, but you're likely going to have a lot of lichens around. I also mentioned Freulania. It's an abundant liverwort, especially on bark, but it also occurs on wood and rocks. They're really difficult to determine the species. And something that's really interesting about Freulania is that picture on the lower left shows these little kind of helmet-like structures inside and on the underside of the leaves. These are called lobules. And, you know, to further advocate for biodiversity, most Freulania have living organisms, a group of organisms called the deloid rotifers that live inside these lobules, right? So you have basically a microorganism that's living inside this micro plant. And I just like to share that with people because it's pretty cool when you think about the diversity. Hornworts, there's approximately 150 hornworts worldwide. Texas has between 13 and 14. It's the least, so it's essentially, it's the least diverse group of bryophytes, and they can be difficult to find. And this is coming from somebody who's, you know, been in the field a lot for the past 20 years, and I've only come across hornworts maybe 5 or 6 times in my life. So they're not as abundant as the other two groups. The next group I'm going to talk about, and the lichens or lichen-forming fungi, lichens together with bryophytes make up that term that Jason used in our introduction, cryptogam, which just really means hidden plants. The lichen-forming fungi are essentially symbiotic entities. There's a fungal partner that's either an ascomycete or basidiomycete fungus with a photosynthetic symbiont, a partner that's, you know, conducting photosynthesis. And that is either a green alga or cyanobacterium. In some cases, there are both green algae and cyanobacteria working together inside a fungus. Over 98% of lichen-forming fungi are of that ascomycete group. However, I am going to talk about basidiomycete lichens towards the end. These are found in most terrestrial habitats, and again, it's why bryophyte and lichen people always hold workshops together. Where you find lichens, you find bryophytes, and there's a huge diversity of secondary compounds called lichen acids. We use those to identify lichens. If you see that piece of sandstone there in the lower right from Palo Pinto County, the different colors are reflective of the different acids. And then that black splotch on the rock, those are our cyanobacterium partnered fungi. They have a cyanobacteria as their photosynthetic partner, and that's what makes them so dark. Nomenclature, when we talk about the species name of a lichen, I'm going to show a lot of species names of lichens. We're naming the fungus. That's the real diversity in lichen-forming fungi. There's about 27 to 30,000 species of lichen fungi that partner up with about 1,100 different algae and or cyanobacteria. There's 6,000 species in North America, just under 1,000 species documented so far for Texas. So this is just a picture of fungi that I've taken over the last couple of years. On the left are, these classic basidiomycete mushrooms, okay? So these are basidiomycetes. The upper right is actually an ascomycete fungus, and it looks like, as you'll see in a moment, a lichen reproductive structure. This is an ascomata, and so we'll talk a little bit about those. On the lower right, those are slime molds that you have. If you have kids, you see them in playgrounds growing on bark, or if you garden, you see them growing on mulch. They're actually not considered to be fungi anymore. They're considered to be a lineage of protist. The thallus of a lichen is comprised of a cortex and an inner layer called the medulla. The algal layer is typically present in that medulla layer. There's sexual reproduction via spores. If it's an ascomycete lichen, which most of them are, they're called ascospores, or they're called basidiospores in the basidiolichens. And there's also lots of methods of asexual reproduction, and I'll mention that in just a moment. The lower left shows an apothecium from an ascomycete lichen, and in that disc-like structure, you'll find spores, the black discs, and the greenish-gray discs of the diploschistes and usnea, respectively, are apothecia that are reproductive structures containing ascospores. The organism on the upper right, the Flavoparmelia, just reproduces asexually, so you don't see any of those sexual reproductive structures, the apothecia. Here's just to kind of just go over this. This is a picture of probably the most common lichen in Texas. It grows on bark. This is Physia apolia. You can see those gray rimmed apothecial discs inside are where the spores are. That gray layer is the cortex. If you were to scrape that off carefully, you'd get to the white medulla. And if you were to look at a cross section, you'd see the gray thin layer, a thick white layer being the medulla. And then between the cortex, the gray layer, and the white medulla, you'd see a bright thin line of algal cells, green algal cells in this case, something that you can do just at home. You can cut one of these with a hand lens or microscope, a dissecting scope, see this for your own eyes, that there's symbiosis going on here. There are three major forms of growth for lichens, and I'm going to briefly talk about those. The crustose lichens. Crustose lichens appear two-dimensional. They're attached directly to the substrate, and they don't have any other, overlapping lobes or structures on the periphery of the organism. So you can see some on the two images on the lower left. You can see some nice discs, those apothecial discs. The Alexoria varia has kind of elongated apothecia, so they don't look like discs. They look like somebody took a little black pen. And then that limey green one or yellow, we always argue about whether it's green or yellow in the lichen world of this one. That has no sexual reproductive structures whatsoever. It's just basically asexual propagules in the form of this yellow dust that has thallus and medulla inside with the algae, and it disperses by wind and water. The next growth form is the Foliose lichen growth form. this refers to leaves, right? So they look like leaves. So if you look at the periphery of these, they have these lobes that are undulating sometimes or overlapping. And so, that's why they refer to them as being foliose or leafy. But there's lots of different subsets of these. The image in the upper right, the Enchelium conglomeratum, is actually a cyanobacterial folio species. So again, the lichen-forming fungi that partner with cyanobacteria tend to be dark brown, slate gray, or black, as is in the case of this one in the upper right. Fruticose, these are the kind of shrubby or bushy or pendulous lichens. So, if you go into the Pacific Northwest, you see these hanging off trees. They're, these pendulous lichens. So these are, really common. The orange taxon on the right of the screen is probably, especially for those of you around Austin, our most common fruticose lichen, and it's very showy, that Teleschistes exilis. In the middle, those cladonia. So, you have basically a foliose type like thallus, but then it has these reproductive structures. And so that's why we call it a fruticose lichen, because it, it has this in 3 dimensions. And then Ramolinus celastrans might be one of our largest lichens, or at least fruticose lichens in the state. And it's also really common around the Austin area. You can find it growing on most bark substrates there. The last cool thing I want to mention before I start getting into talking about methods and how you go about researching these organisms are that lichens are really micro ecosystems. It's just amazing. So when we're collecting lichens, we're also collecting fungi that are parasitic specifically on lichen-forming fungi. So this picture is a picture of a foliose lichen, Pyxine subcinerea, with little tiny black apothecial discs of a fungus that's parasitic to that specific species. Globally, including Texas, there's very little known about these taxa with respect to their diversity and their host specificity. Endophytic fungi. When we haven't gotten very cold yet. So when you leave your office space, your house, wherever you are, and you grab a leaf, whether it be a grass blade or a tree leaf, very likely there are fungi, not parasitic, sometimes considered innocuous, perhaps beneficial at times, and we call these endophytic fungi. They've recently discovered that a typical lichen thallus might have between 10 and 20 different species of fungus growing within that thallus, and they don't know much about their roles yet at all. And lastly, and just like mosses, you can find a lot of invertebrates living on and not inside the organisms, but within the webbing of the thallus and the structure. So similar to mosses, you can get these wet and wring them out and find water bears and nematodes and all sorts of ciliate protists and rotifers and things. So they're really a micro ecosystem too, which is something that's not discussed often. Recently, just to kind of, talk about a recent piece of work that we did in Montague County, which borders the Red River and therefore Oklahoma. We worked on about 150 acre property. We found 49 bryophytes. So this included a hornwort, a handful of liverworts, and the rest being mosses, about 42 species of mosses, and 180 lichens and allied fungi. What do we mean by allied fungi? Why those lichenicolous fungi? We identified several lichenicolous fungi, the parasites on the lichens. And then there's also some fungi that are truly free living. They don't have algae, but as lichenologists, they're showy enough to where we collect them, and a lot of times we include them in biodiversity surveys. One of the cool results of this, besides just the sheer number of species, there were 430 vascular plants found in this property. So almost, that's 50% almost of the diversity are cryptogams, bryophytes, and lichens. Is that table 1 are new lichen records, just lichen records for the state of Texas. So, we recorded 30 taxa of lichens that hadn't been recorded for the state. We also only made about 5 trips there. So if we continued to go back to this place, we would undoubtedly add to those lists. Over the past 4 years, I've been collecting in these sites here. They include several state park sites, TPWD sites. I would say that Fawcett Wildlife Management Area is the one that I've been spending the most time in with respect to a Parks and Wildlife site. I've also been spending a lot of time collecting in Tarrant County, where I live. I have permits from the city of Arlington and the city of Fort Worth, and so been making a lot of headway there, and I'll talk a little bit about some results coming up here soon. When we conduct field work, it's the same between lichens and bryophytes because they co-occur. So, you go out with a hammer and your chisels, and you're collecting off of bark and wood and rock, and sometimes they occur directly on the soil. I collect with little tiny paper bags, and you just slip in a collection number with each specimen. And so the collection techniques are really similar as our substrates. Again, the substrates where mosses occur, you find lichens as well. Processing bryophyte specimens. Bryophytes, so the mosses, liverworts, and hornworts are pretty easy. We just make these packets, whether they be temporary or permanent when they go to a museum or university to be stored. But you make these temporary packets. I use two different colors. I use white for the original collection, and then yellow packets for the duplicates, and you number them, and then you have a little piece of paper or somewhere writing what substrate it grows on. We enter all this into a database, or I do right after I collect them, even though I haven't identified stuff yet, because that's really important with getting labels made. Identifying bryophytes, I'm just going to focus just talking on mosses right now. So you have to have a compound microscope as well as a dissecting microscope. And I will say I identify lichens, and I work with collaborators who identify mosses. I have identified mosses. It's very challenging. We look at the leaf structure and anatomy. You look at that picture of the blown up of the capsule with the little teeth around it. You look at sometimes you have to look at the reproductive structures, and a lot of times you can't find mosses with the reproductive structures. These are really challenging organisms to identify, and also to find really, good keys with good terminology to work your way through them. So with respect to lichen specimens, they're processed a little bit differently. I follow the way I was trained in my graduate program by Bob Egan, who taught us lichenology. We use cards to put the specimens on. Sometimes, like the Sora crenata, that white lichen on the lower left, you can just put them on a piece of fabric and put them on a card and into a packet. But most of the specimens, if they are on wood, bark, or rock, you glue them. I glue them to a card and weigh them down, and then you put them into those numbered packets and make a spreadsheet with all of your data to make labels. Identifying lichens, I'm going to focus a little bit more on this because this is what I do on a daily basis. You need to know a lot about the morphology, the gross morphology, the anatomy. So a lot of times you need to have a compound scope to identify these as well. The chemistry is very important, and that can be kind of tricky. And then now, of course, we're using DNA to identify and other molecules to identify these organisms. So, what growth form is the lichen? What type of apothecium does it have? Or does it have these asexual propagules? The asexual propagules are little fragments that break off, and then if they fall in the right substrate, they can grow into a clone of that organism. And so, a lot of this gross morphology you can see with a hand lens, but a lot of it you have to take back into a lab and work on it. Anatomy. So I took this from a recent, well, not a recent, but a paper that looked at ascospore diversity on the right of the screen. And so it shows you all these different spores that shape and size, and sometimes, like in the upper left, each spore is one cell. Some of the cells are two. Some spores are big, and they're made up of hundreds of cells. And so there's a huge diversity of cells, of ascospores, the size, the shape, the ornamentation, and you have to have a scope to look at that and also be able to measure them. The lower left is just a picture of some algal cells with some red stained fungal hyphae surrounding it. Sometimes, not common, but sometimes you need to know whether the green algal partner is a Trabuxia type, like this kind of spherical shape, or is it a Trentipolia type, which is a more kind of elongated type of structured green algal type. So, you know, again, there's a lot of terminology that and background that goes into just really being able to talk about these organisms. Chemistry is super important. You really can't do basic lichen work. So I have a picture of the three little bottles I use the most. One of them, the K bottle, is 10% potassium hydroxide. I use a lot of just water, right? And then the C is Clorox for bleach. And so the C and the K are really important because there's so many different lichen acids, and decades ago it was found that those chemicals react with these different lichens acids to give these color code profiles. However, sometimes you have specimens that are UV fluorescent, and so you need a UV cabinet. That pyxene, that gray in a UV cabinet, I took this picture with my cell phone, it's just bright yellow gold in the UV cabinet. And then we do a lot of thin layer chromatography. And so this is a profile of several different species in the genus Parmotrema. And so you really need to have all these tools available to you to identify lichens properly. A lot of people have been working with me on these projects. Folks at the California Academy of Sciences have been helping with mosses, and just these other people are you know, pivotal in because you can't collect thousands of specimens and do all the work on your own, especially when I don't specialize in bryophyte identification. So I just wanted to point out that I, you know, work with a group of people and have support from folks like the Texas Parks and Wildlife Division and the Arlington and Fort Worth Parks. So I just want to briefly provide just some results because I just don't have a lot of time to do it all. But so, for example, Fawcett Wildlife Management Area, I've collected 766 specimens of bryophytes and lichen. That includes lots of duplicate specimens, and it also includes lots of, you know, unique specimens. So far, we've identified 89 bryophyte species and 147 lichens. At Fort Boggy State Park, you can see the numbers there. And the point being that, you know, there's a strong correlation between your effort and the diversity of which that will reflect. The other place that I've spent a tremendous amount of time, because some of these sites are within walking distance from my house, are Arlington City Parks. And so far we have 78 bryophyte species, including a number of new species to Texas, new mosses that have not been recorded for Texas. They're not undescribed species, they just haven't been reported for Texas yet, and 122 lichens from the city of Arlington's park system. So just for example, this slide shows some under-collected mosses that we've reported from just Tarrant County. The taxa that have asterisks after them are taxa that have not been reported for the state of Texas yet. And so we're going to be publishing a bryophyte record for Tarrant County soon, and that will reflect those new species being reported for the state. And then some of these others, like the Physidens crispus, that has one record in West Texas. So now it has two collections from the state of Texas historically. Specifically, one species, Venturiella sinensis. If you look at the map of Texas, the blue asterisk there in West Texas is where it was reported prior to 2017, and we've picked it up in 5 counties, and we expect it throughout North Central Texas and in the Panhandle. Similarly, Lorenztiella imbricata had many, many more records than that Venturiella species, but if you see here, the blue is where it was previously collected, including a collection from extreme South Texas, but now we have reports from, several reports from North Central Texas. Really, really neat moss. It's just shiny silvery, and the sporophytes are inside that little ball of kind of silver wool. Rixia, I told Rixia is the most abundant liverwort. I've collected several species of Rixia, and I've only had a couple identified. There's one person in North America at the Smithsonian Institution who's really able to identify Rixia, and you know, that it's just, sometimes it's not plausible to get everything with a name on it, especially if you want names on them in a timely fashion. We've uncovered lots of cool liverworts. This Spherocarpus texanus. I mean, some of these specimens only appear when the soils are really moist. So, you know, you got to get out when it's wet and the weather's cool. So we found a lot of these at the Fawcett Wildlife Management Area in Palo Pinto County. It was early March. There had been some rain, and it was really cool. And so that's when you find a lot of these liverworts. They tend to be ephemeral in their presence. Freelonia, that leafy liverwort. I just wanted to talk a little bit more about that because I've made over 150 collections of Freelonia at the sites that I've been working at. We got 2 collections of Freelonia appalachiana from Fort Boggy State Park in Leon County, that's south of Centerville, and they're new records for Texas. Freelonia virginica from Purvis Creek State Park makes the 2nd collection in the state. We have several collections from Fort Worth of a undescribed taxon, which has also been collected. It's in Oklahoma and Missouri, but John Atwood is in the process of naming that species. And then I listed the other species that we've collected. The picture of Freelonia inflata, I cited where I got it. I didn't take that picture myself, but that's kind of the weed of Freelonia. So 9 out of 10 times you see one of these green Freelonias growing on bark, it's going to be that species here in north central Texas. But we do, as you can see, have other diversity here. This is from a paper that John and I published in 2019, and these are 6 species and their distributions in northwestern Missouri, and this is what we're working on for a publication for north central Texas. So this should be coming here in the next year or so, but we feel like we want to go out and make some more collections to see if we can, continue to illustrate more diversity in this part of the state. Hornworts. I've collected 1 hornwort in Texas. It was in Mason County. It was last spring. Hornworts are really interesting. They're thought to be, really the most ancient lineage of plant. And so the lower left-hand corner is a picture of the thallus, and then on the right you'll see the horn. Right, that's the sporophyte. That's the structure that produces the spores. And what I have in the middle is yet another stellar picture that I took with my old cell phone. Hornworts are the only lineage of plants that have a single chloroplast in each cell. So talk about conformation of having a hornwort. That's one large chloroplast in each cell, and you don't see that with any other lineage of plants. And so I just like sharing that because it's one of the characters that makes hornworts a unique evolutionary lineage. With respect to lichen biogeography, I have a couple of pictures of these 2 specimens on the right that we collected at Mission Tejas State Park and nowhere else. And East Texas really reflects the southeastern United States. South Texas, reflects that northwestern part of Mexico, and similarly, West Texas reflects the Chihuahuan and also Sonoran Desert influence. And so you have similar biogeographical patterns that you find with vascular plants and bryophytes. And you also have species that are very widespread. And similarly, we have vascular plants that have those distributions. Just wrapping up some of the things that we're working on. Parmotrema is one of the most diverse lichen taxa in the world. I think there's over 300 species. There's a lot of species in Texas. I've listed the ones that we've collected so far in north central Texas. The 2 with the asterisk, Dyspectum and Parmotrema dyspectum and Parmotrema eciliatum, are new records for the state of Texas. We also have a taxon that we think it's a new species. Now, these differ in morphology and chemistry, but we also need to use some other tools. And so we're proposing to use a technique called RAD-seq. I'm not going to go into it. It's a means of extracting a lot of DNA to elucidate relationships and to place difficult taxa where they belong. This is a group, the Punctelia graminicola group. It includes 3 species. They differ in chemistry, and they also differ in these asexual spore-like structures called conidia. And I've really never been convinced that we have 3 distinct species. And so we're also going to be using RadSeq to elucidate relationships there. This is a really cool find from Fort Boggy State Park. So I took this picture of this lichen. It's a basidiomycete lichen. And I thought for sure we had found Multiclavula vernalis, basidiomycete lichen that has not only not been reported for Texas, but a basidiomycete lichen had never been reported for Texas. On the lower right is a picture from California that I took off the web. You can see this site. It was growing on these kind of muddy shelves, but I wanted to make sure that it was multiclavula, so I sent some of the fruiting bodies to my friend Todd at the Field Museum. He's a phylogenetics expert, and we found out that when we looked at the DNA that it didn't come out as a multiclavula. It came out in the genus Bryoclavula that was just named in Japan in 2020. So Bryoclavula phycophylla was named in 2020 by Hiroshi Matsumoto. And, I'm not going to really talk about these trees, but what I can show you is the tree on the right is the tree that Todd produced with our material. And, you have in the green box up top multiclavula, and way down lower you have the now two species of Bryoclavula, and we're publishing that in the Bryologist, and that should be out soon. So here are close-up pictures of the fruiting bodies. They kind of, as they age, they turn brown at the tips, and the tips kind of start to taper. Again, this is this picture of the algae being surrounded by fungal hyphae. One of the evidences that it's actually lichenized. And then some other bodies that, again, a term that I didn't know very well before this, but this is a sterigmata. It's a sterigma on the lower right. It's a structure that holds, you can see 6 spores at the end of it. With respect to Mason County, I started collecting in Mason County when I was in graduate school, but I've recently gone back. So far, we have 173 lichen species, and that's just from what I've collected, and about 65 bryophytes. We think that number of lichens may approach 300 species. Caleb Morrison, Kansas, also is collected at the Mason Mountain Wildlife Management Area, and he's put names on that. Mason County is just so cool because of the geological diversity. And so that leads to lichen and bryophyte diversity as well, right? Substrates. Future work, besides finishing these current projects, the Texas Panhandle is just unknown with respect to lichen and bryophyte diversity. There's some great sites to go there and collect, as well as continuing to make connections with private landowners. The high elevation sites in West Texas, Davis Mountains and some of these TPWD properties, they make excellent baseline, really foundational work for environmental change studies. These climates are, these environments are affected, severely by changes in temperature. And I really think it'd be important to go out to these sites high up in West Texas and get some baseline data. And then the Edwards Plateau, it's just a dream of mine to do a project on the lichens of the Edwards Plateau, and of course collect bryophytes and make some discoveries there, but I'd really like to work on the Edwards Plateau.